Our next speaker, Rama Shivakumar, is the Manager of Technical Applications at MaxCyte.
Hello, my name is Rama Shivakumar and I am the Manager of Technical Applications at Maxlite Incorporated. In this talk, I will discuss the clinical scale gene editing for cell and gene therapy applications. The talk is organized in the following segments. We will first go over who we are as a company and how our technology has provided the enabling solution to our customers' research and clinical needs. To illustrate this, we have included 4 specific case studies. In the first case studies, we will go over the optimization of research scale gene editing in Ipscs. In the second case study, we will illustrate ways in which to improve the efficacy of CAR-TCR therapies with gene editing. In the third and fourth case studies, we will talk about the manufacture of a gene-edited cell therapy product for the preclinical and clinical application in hemoglobinopathies like sickle cell disease and beta thalassemia. We will then round off the talk with a summary. Let us first go over who we are and how we are driving the next generation of cell-based medicine. MaxCyte is a global cell therapy and life sciences company that was founded in 1999. We are headquartered in Gaithersburg, Maryland. MaxCyte's powerful, scalable flow electroporation technology is currently being used to engineer a variety of cell types to enable research, clinical, and commercial applications. Once the transfection process is optimized at the research stage, the high-efficiency, reproducible performance can be translated to a clinical or commercial scale rapidly without the need for re-optimization. The same unifying technology can therefore take a product from concept to clinic. MaxCyte's electroporation platform is branded as Expert. The Expert platform is designed to deliver high transfection efficiencies and viabilities to give you a potent cell therapy product that is efficacious and reproducible, which can then enable the rapid clinical translation of your research to a therapy. The Expert is a family of 3 instruments: ATX, STX, and the GTX. All these instruments are sleek, with a touchscreen and enhanced software user interface occupying very little space on the lab bench. The ATX is research-focused, used for small and medium-scale static transfections. The STX is a scalable flow electroporation device that is used for transient protein production and also in the production of vaccines and other biologics. The GTX is the clinically validated scalable flow electroporation device which is 21 CFR enabled, has its cGMP compliance and also an ISO certification. We offer many consumables that are compatible with the Expert devices. These are called processing assemblies, and one can use them to transfect a range of 75,000 cells to over 20 billion cells. The transfection volumes range from as low as 15 microliters of cells in the smallest chamber to up to 100 mils of cells in our flow electroporation chamber. With these instruments and processing assemblies, we have assembled a number of partners like KSQ, Sangamo, UPenn, CRISPR Therapeutics, Allogene, and Kite Gilead, to name a few, to scale very quickly from R&D to patient treatment. We have over 120 clinical licenses, are engaged in over 90 clinical programs, and have over 11 commercial licenses. In the first case study, we will talk about gene editing in patient-derived Ipscs. We will show work that was done in Dr. Hotta's lab at Saira in Japan. The goals illustrated in this case study were to optimize the electroporation conditions in Ipscs for disease modeling. The Lonza electroporator and the MaxCyte platform were compared side by side in the disease modeling experiments outlined. CRISPR RNPs and an ssODN template targeting the Duchenne muscular dystrophy gene were electroporated into patient-derived Ipscs to induce exon skipping. Knock inefficiencies and cytotoxicities were then assessed. The MaxCyte platform was used to optimize electroporation conditions into patient-derived Ipscs. GFP mRNA was electroporated using protocols delivering low, medium, and high energies. 100% transfection efficiency was achieved in all conditions, with the MFIs increasing with the increasing energy delivered. The MaxCyte platform was used to electroporate RNPs and increasing amounts of ssODN knock-in template to induce exon skipping by disruption of the splice acceptor site on exon 45 of the Duchenne muscular dystrophy gene. When the high-energy protocol was used, over 60% knock-in efficiency was observed, as evidenced by the introduction of the AGE1 site. Higher, consistent knock-in efficiencies were observed when using the optimized protocols on the MaxCyte platform when compared to other commercially available electroporation systems. The MaxCyte system also demonstrated better recoveries and reduced cytotoxicities. In our second case study, we will go over ways in which to improve the efficacy of CAR TCR therapies with gene editing and how this technology enables the making of a more potent immunotherapy product. The work done here was conducted at Dr. Carl June's lab at UPenn. Genome editing, when combined with TCR immunotherapy, has the potential to increase the potency and safety of the engineered T cells. The Phase 1 clinical trial, illustrated here, used the MaxCyte GT platform to electroporate patient T cells with CRISPR-Cas9 RNPs containing 3 single-guide RNAs targeting the TRAC, TRB, and the PD-1 loci. The edited T cells were then transduced with a lentivirus expressing a TCR specific for the New York ESO1 tumor antigen. The engineered T cell product was then infused into the patients. The engraftment and persistence of the edited T cells in the patient were monitored over time. Autologous T cells from 2 myeloma patients and 1 sarcoma patient were obtained by apheresis, elutriated, washed, and enriched for T lymphocytes. Electroporation of the RNP complexes containing equimolar mixtures of Cas9 with guide RNAs for the TRAC, TRB, and PD-1 were performed as per MaxCyte's standard operating procedures. The RNP complexes were mixed with the desired concentration of T cells and loaded carefully into a processing assembly and then electroporated using an optimized protocol on the MaxCyte GT instrument. The cells were then cultured in the appropriate media with IL-7 and IL-15 at 30 degrees centigrade for a time. Following the gene editing step, the cells were transferred to a 37 degrees incubator activated with CD3-CD28 beads and transduced with a lentiviral vector encoding the TCR specific for New York ESO1 antigen. The cells were subsequently expanded in a wave bioreactor with IL-7 and IL-15 until harvest day. The final genetically engineered T-cell product was termed as NICE, which referred to the New York ESO1 transduced XXX gene-edited cells. At the end of the 8 to 12-day expansion protocol, over 1E10 cells were obtained. Digital PCR was used to determine the gene editing efficiency. The results varied depending on the guide RNA used. Gene editing efficiency for the TRAC locus was 45%, for TRB locus was 15%, and for PD1 was 20%. At the ASGCT meeting in 2020, Dr. Carl June affirmed that the gene editing reagents have come a long way from the first generation reagents that were used in this study. They are now getting over 90% gene editing efficiency for the 3 loci as they embark on expanding this trial to include a larger cohort of patients. The high fidelity on-target editing for the TRAC and PD1 locus was well over 99%. It was a little lower for TRB, but then the gene editing efficiency was lower for this locus as well. When the NICE cells were co-cultured with the target cells expressing the New York ESO1 tumor antigen in an in vitro cytotoxicity assay, the NICE cells demonstrated potent antigen-specific cytotoxicity over a wide range of effector-to-target ratios. In fact, the NICE cells showed an even greater cytotoxicity than the control cells that expressed the transgenic TCR but were electroporated without the CRISPR reagents. In another assay to assess the polyfunctional profile, the NICE cells that were activated with the CD3-CD28 beads secreted very high levels of interferon gamma in response to the target cells. The patient outcomes after the infusion of the NICE cells were encouraging. The frequencies of the gene-edited and TCR-engineered cells remained quite stable between day 10 and 4 months post-infusion. There was also an increase in the expression of genes associated with the central memory phenotype. Samples were collected from patients between 3 and 9 months post-infusion and expanded in the presence of New York ESO1 antigen. Antigen-specific cytotoxicity was assessed by chromium release assays and was observed in all the patients. The CT scan from one patient also showed a 50% regression of a large abdominal mass. In the third case study, we demonstrate the cGMP scale manufacturing of the gene-edited cell therapy product for the treatment of sickle cell disease. The MaxCyte GT platform was used to electroporate patient-derived SCD CD34 positive cells with the SCD mutation targeting single guide RNA, Cas9 protein, and an SSODN template carrying the intact beta-globin DNA sequence. The durability of the gene-edited cells were assessed in an in vitro colony-forming assay as well as engrafted into mouse models for in vivo studies. The Phase 1 cGMP manufacturing outcomes were promising with excellent viabilities of 81%, robust colony-forming unit formation, and gene correction efficiencies of 26%. The in vivo studies in the mouse models showed a very high level of engraftment of about 88% with the persistence of the SCD edited cells. In the last case study, we demonstrate the feasibility of CRISPR-Cas9 gene editing using the MaxCyte platform to treat sickle cell disease and transfusion-dependent beta thalassemia in a clinical trial. The cell therapy product was manufactured using the MaxCyte GT platform where the autologous patient-derived CD34 positive cells were genetically edited at a clinical scale using CRISPR-Cas9 to knock out the BCL11A erythroid enhancer and reactivate the production of fetal hemoglobin in 2 patients. The patients, one suffering from SCD and the other suffering from TDT, were monitored for assessments of gene editing efficiencies, engraftment of the edited cells, and clinical outcomes. For the TDT patient, levels of fetal hemoglobin increased rapidly from 0.3 grams per dl at baseline to 8.4 grams per dl at month 3, 12.4 grams per dl at month 12 to 13.1 grams per dl at month 18. The percentages of red blood cells expressing fetal hemoglobin increased from 10.1 at baseline to 99.7% at 6 months and was maintained through month 18. For the sickle cell disease patient, the fetal hemoglobin fractionation increased from 9.1% at baseline to 43% at month 15. The percentages of red blood cells expressing fetal hemoglobin increased from 33.9% at baseline to 99.9% at 5 months and was maintained through month 15. The case studies illustrate the diverse applications of this technology in the gene editing space, from research applications to the generation of a commercially viable cell therapy product. The non-viral approach discussed here is robust, safe, and cost-effective. The consistent, non-toxic gene editing demonstrated in Ipscs can be used for disease modeling and potential therapeutic applications. This technology is also used for the cGMP manufacturing of a potent cell therapy product for the treatment of hemoglobinopathies like sickle cell disease and transfusion-dependent thalassemia. Gene editing with the MaxCyte platform can not only be used to generate an allogeneic cell therapy product, but also to improve the efficacy of an already existing immunotherapy model. At MaxCyte, we have a partnership approach to our science. We have a globally distributed, goal-oriented field applications and sales team. We have a very experienced in-house R&D team. We also have a skilled quality and regulatory affairs team who handle our FDA master file and quality documentation. The expert platform is more than just a technology. It is a solution. Thank you for listening and for your patience. I can now take your questions.
All right. Thank you so much, Rama. Really appreciate the talk. And thanks to the audience for attending this session, the overall summit. We're going to go ahead and we're going to transition now into some Q&A for Rama. If you haven't had a chance yet, if you could go ahead and you have a question, go and look on your left side. There's a question mark icon. Click on that to open up the Q&A box. And again, please feel free to leave a question if you have one. Let's take a look right now. What do we have? So we've got one question from the audience so far. Rama, how do you think electroporation technology will improve in the next 3 to 5 years? What do you think is driving these improvements?
I think most electroporation technologies are looking to be in the clinic in the 3 to 5 years, like we are. Certainly most of our competitors are. As the field moves towards allogeneic therapies, you'll have a lot more gene editing applications becoming important in the clinic, and to deliver the gene editing tools, electroporation becomes a big factor. So I think most electroporation technologies will have to be cGMP certified,
they'll have to be closed, and also like kind of have this integrated workflow that integrates the upstream and downstream processes in a smooth workflow. So that's where I see that's where I see this technology heading.
Awesome, really awesome answer. Second question for folks is why do viral versus non-viral gene editing? So could you kind of expand on that a little bit, please?
Why go through non-viral gene editing?
I think that's what the question is asking.
Well, I think for obvious reasons. I mean, viral gene editing, first of all, just making the gene editing tools into packaging them into viral vectors is a laborious process. And secondly, viral vectors are known to have these, insertional mutagenesis, which can be completely avoided with non-viral gene editing. And it's just a lot easier to go through non-viral gene editing because of the editing tools that are available as either RNPs or RNA that we can deliver quite easily through electroporation.
All right, makes perfect sense. Not seeing any additional questions yet. Let me go ahead and ask one from my own end, just listening to your talk. I noticed in one of the case studies, I think it was one with a collaborator at UCLA, you had a pretty robust HDR integration. I think it was like 26% or so. Mm-hmm. What do you think, where do you see the field at this point? Because I know that's something people are looking to improve those numbers. Why did you get such a high integration, and how would you recommend, or how have you seen your collaborators improve HDR integration?
I think there are many ways you can improve HDR integration, that people are looking at HDR enhancers and other things. But in this particular case study, I think they did get pretty high HDR through the single-stranded SSODN knock-ins, which we saw with our other collaborators in NIAID, like John Tisdale. So I think the next step for them would be to, they already have looked at mouse models, and they're going into clinic with this, but John Tisdale is also looking at, with us, looking at non-human primate models to see how the engraftment goes, and to improve the engraftment efficiencies is where we're headed now.
Okay, gotcha. That makes plenty of sense. Let's see here if there are any other questions from the audience. Let's see here. Not seeing any other. So folks, if you have any other questions, please put them in the chat. One thing that I wanted to bring up is right now GenScript and MaxCyte are going through a nice collaboration where we are exchanging materials and reagents to work to modify immune cells using single-stranded DNA. I know, Rama, that your team is kind of cursory attached to that project, but just wanted to bring it up for folks and let everyone know that we look forward to presenting some preliminary results with your team at ESGCT a little later in the year.
Yes, certainly. I think all that data is being pulled together right now, and I think there may be some opportunities for a future talk where we can present that data as well.
Sounds great. So one more question from the audience so far. Why is the Maxlite system superior to other systems on the market? Little pointed. Could you kind of talk a little?
Well, yeah, it's kind of hard to answer that question without coming off as a sales pitch, but I can say that the first reasons, as I mentioned from the first question, is we are a scalable electroporation platform. So as what I talked about in some of the case studies that we presented, we can function at a very small scale, which is about 50,000 cells, as we presented in the IPS case study at a research scale. And then we can go all the way to a clinical scale where we can electroporate up to 50 billion cells. And so that is one of the biggest attractions that most of our customers come to us for. And we also have a cGMP clearance and 21 CFR compliance and a user friendly interface on our machines, and a number of different processing assemblies that can be used for different applications depending on the scale that you're looking for. And one of the biggest attributes of the system, as I mentioned in the talk, is once the optimization is done at a small scale, you don't really have to re-optimize as you scale up, and you get pretty much reproducible, consistent data when you scale up for many applications, as we talked about in gene editing applications, which is both knockouts or knock-ins that I showed in this presentation.
Very cool. And then it looks like another question from the audience. Have you compared with a company called Oncosec, something like that, ONCOSEC?
You mean gone on a head-to-head comparison? No, I have not heard of that company, actually.
Okay, got it, yeah.
So, yeah.
Not entirely sure. We'll see if there's a correction or another comment from that attendee. Let's see here. Any other questions from the audience? It does not look like it so far here. So let us go ahead and let's close out the session at this point. Rama, really appreciate you presenting today. Really enjoy the talk and the collaboration. And let's go ahead and I want to thank the audience as well for your time attending this talk and then the track and the summit. Just one housekeeping note before we close things up here. This session and this track is going to close at 5 p.m. We're going to go ahead and have a closing keynote talking about the second generation of CAR T-cell therapies. In the meantime, please feel free to go into the exhibit hall, attend the booths, and everything else. And again, hope everyone has a great rest of your day.
Thank you so much.
Thank you much, Rama. Appreciate it.
Bye. Bye.
Bye-bye.